So for meiosis itself, let's go through some of the details. It's going to have the same general stages as mitosis, so I don't want to go through everything in painful detail when we've already covered a lot of this. So let's just make sure we're clear. With mitosis, we went through one division, which meant we got two cells that were each 2n. They were copies. This is going to be used for asexual reproduction because it is great at producing identical copies, clones. We want that when we're going, for instance, from a zygote to us, because we want all of our cells to be an exact copy. That's useful for us. When we go through meiosis, we're going to do two divisions. So when we're going through this process of meiosis, you'll see mitosis here, we copied our DNA, then we went through and we divided and we got two cells, each that looked identical to the start. But for meiosis, you'll see we're going to do two divisions. So we've got division 1 followed by division 2. And so this two divisions doesn't give us two cells, it gives us a total of 4 cells. But we do not, I repeat, we do not copy our DNA between the two divisions. So we essentially copy our DNA so you can think of this cell as being 4n, right? Then we divide it, we get two 2n's. That seems to work out nicely. But when we're doing meiosis, because we don't copy our DNA again, when we first divide our cells, we're effectively getting down to being like 2n, all right, each, just like in mitosis. But then we copy them again, which gets us down to being 4 1n cells. This is one of the differences. Because we didn't copy it a second time, each cell essentially gets half of the normal amount. Instead of 2 sets, we're down to 1 set. That's why we get gametes. That's why it produces 4 1n cells, 4 haploid cells. So this means it's very useful for sexual reproduction because in sexual reproduction, we're trying to make different offspring genetically not like the parent. And to do that, what we can do is combine two separate individuals' gametes. So if I produce sperm and somebody else produces the ovum, they don't have the same genetics as me. So by combining our two gametes to make a new organism, we make sure that that organism is not identical to the female. It's not identical to me as the male. It's a new combination. This is great for diversity, which is good evolutionarily. So that's the purpose of this in meiosis. Two divisions, haploid cells, you produce 4 of them though. So the major differences that we're going to talk about that occur during this process, and you'll see a lot of these are important because they give us this variation as well, is when we go through meiosis 1. So meiosis 1 is going to be this kind of first division here. So meiosis 1, when we do that, we're going to do where the homologs pair up during prophase, and they're going to undergo a process called crossing over that we'll discuss on the next slide a bit more. But it means that these homologs, because they're equivalent, they have the same genes on them, they can actually swap pieces of themselves. So that means a chromosome will no longer have to be just mom's chromosome or just dad's chromosome. It can be a mix of mom's and dad's chromosomes. This gives us more variation. So this will occur during prophase 1. In metaphase 1, we're going to line up the homologs. Notice these guys are not lining up single file. You're seeing that this homolog from mom is lining up with the homolog from dad that's right behind it. You'll also notice they've undergone crossing over, so some of them are kind of a mix of moms and dads. This is different than how it occurs in mitosis. So in metaphase 1, they line up with their homologs, and in anaphase 1, they separate by homologs. They don't separate the chromatids yet. Notice over here, they still have the sister chromatids attached. Now, crossing overs occurred, so they're not exactly all moms or exactly all dads, but they still have undergone this process where the homologs are still together. So with meiosis 2, just to make sure we're clear, there is no DNA replication prior. So there's no DNA replication right here. This is meiosis 2. And so because of that, when we go through and divide this, you'll see we start off with half the normal amount. So if this is mitosis, we would have started out with 4 sets of sister chromatids. Instead, we only have 2. And this means we're going to end up with, instead of getting 4 chromosomes like we normally would, we're going to end up with 2. So instead of having us get 2 cells that have the normal amount, we're going to get 4 cells that have half the normal amount. 4 1n haploid cells. That's the only difference between meiosis 2 and mitosis, is the fact we did not copy the DNA prior, and so we end up with half the normal amount we'd had if we did mitosis. Otherwise, we line things up single file, we separate the sister chromatids, everything else is identical to mitosis. So the importance of meiosis is about getting us this variation. That's the benefit of meiosis, that's the benefit of sexual reproduction. So if we're going to get these varied haploid gametes, these varied sex cells that we can use to make varied offspring, I want to make sure you're clear on where that variation came from. So the first part is crossing over, where we can go from having our homologs that are either moms or dads to getting ones that are a mix of moms and dads. So this allows us to get new combinations of genes because we're mixing up our chromosomes a bit. That gives us variation. Next, we've got what's called independent assortment. And this is where when you go through this process of metaphase and anaphase one, where you line up the homologs and you grab one of the homologs, you can either grab mom's or dad's homolog. And because this occurs randomly, there's all kinds of different possibilities of how you can grab them. It's a really high number. You can actually calculate it. If you want to do this, it's going to be how many options do you have. In our case, there's 2, moms or dads. And how many times are you doing it? In this case, it's 23. So from independent assortment, just from this idea of for chromosome 1, do I get moms or dads? For chromosome 2, do I get moms or dads? Just by being able to have all moms, or moms, 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 dads, moms, moms, just being able to do this, you can get about 8 million different combinations of chromosomes. So if you're a male, you can have 8 million different sperm, ignoring crossing over, just by having where the chromosomes moms versus dads differ. Another way of thinking this is imagine if you had a phone number that just has 1s or 0s. Those are the only options. But it's 23 numbers long. So it can be, 1, 1, 1, 1, 1, 1, 0, 1, 1, 1, 0, blah, blah, blah. You could have 8 million different phone numbers in that system because there's 23 options, of which you can have a 1 or 0 for each. There's 8 million possibilities. That's essentially how independent assortment works. Now, independent assortment gives you a lot more variation the larger the number of chromosomes you possess. If you're a fruit fly with only 4 chromosomes, not as many options here. We're only talking about like 16 different possibilities. But we still have crossing over. I mean, that still helps out too. And even after crossing over and independent assortment have given us far more than 8 million possibilities, because this 8 million ignores crossing over. We then get random fertilization, which is the fact that it's not just you that's doing this, somebody else is doing this too. So if we ignore crossing over, just to give you some simple numbers to try to make this more real, there's a 1 in 8 million chance if I don't do crossing over that I would produce identical sperm. The female also has a 1 in 8 million chance she'd produce identical ovum. So we now have to have where my identical sperm, which is very rare, has to somehow find the identical ovum, this has to happen twice, of the female, which is also a 1 in 8 million chance. So to figure out the odds of that, you just have to multiply the 8 million chance for me to produce identical sperm times the 8 million chance for her to produce identical ovum, and you get a 1 in 64 trillion chance that we would produce identical offspring that aren't identical twins. Which identical twins are weird because they came from the same egg and the same sperm, so you can't really include them with this. But there's a 1 in 64 trillion chance, ignoring crossing over, which makes this astronomically higher, that you could ever have identical offspring with the same mother and the same father because of the variation, in humans at least, caused by meiosis. For other organisms, that number can go up or that number can go down a bit depending upon how many different types of chromosomes they have, because that influences independent assortment. But the number's always going to be a big number because of crossing over, random fertilization, and independent assortment. It's always going to be exceedingly, exceedingly rare that you'd ever get identical offspring via sexual reproduction.